Hi everyone, my name is Alisia Tumac, and today I will present the role of immunoadjuvants in immune system enhancement. And I want to start by providing an overview of what immunomodulation is, how it can influence the immune system, vaccination, the different types, and the different components inside vaccines. And then I will go into adjuvants, what are the pros and cons, the timeline of adjuvant discovery, their expected immune response. And then I will go into a couple of specific adjuvants, and I will finish off with a clinical trial overview. So I want to begin by talking about immunomodulation. This is a process by which you can either suppress or stimulate the immune system. Immunosuppressors typically consist of drugs that will produce an unwanted outcome as immunosuppression, such as chemotherapy. Chemotherapy it's highly cytotoxic, and it destroys the bone marrow, which is responsible for producing erythrocytes and leukocytes, and it therefore inhibits proliferation of immune cells as a side effect. But you can also have immunosuppression as a wanted outcome in the case of anti-rejection drugs. Whenever a patient receives an organ transplant, you would administer them anti-rejection drugs to suppress their immune system and ensure that the immune cells will not attack the newly transplanted organ. Next, I will talk about immunostimulants. Immunostimulants can be either specific, such as vaccines, or can be non-specific, such as adjuvants. And I will talk more about in detail about this, but whenever you combine non-specific adjuvants into the specific immune response, you can enhance the immunostimulation as an effect. So what are vaccines? I want to talk about the different types of vaccines. There are in fact 5 types. The first type deals with live attenuated vaccines, such as in the case of varicella or chickenpox. And how would you attenuate the vaccine? You would passage it a couple hundred times through hosts such as chick embryos, and you would do it as many times as you need until you render the pathogen unable to replicate in a human host. However, with live attenuated vaccines, you have the danger of the virus reverting back into a virulent strain, such as in the case of the polio vaccine. And researchers have therefore come up with a second type of vaccine, which is inactivated or killed type, such as in the case of polio or hepatitis A. And so, because most pathogens are made up of proteins or DNA, RNA, you would typically use heat or chemicals such as formaldehyde to inactivate these pathogens. The third type came up, and it's the toxoid vaccine type, such as in the case of tetanus vaccine. And it basically immunizes patients against not necessarily the pathogen, but rather the toxins released by the pathogen, such as the tetanospasmin toxin. And then the fourth type of vaccines is encountered in the pertussis or whooping cough vaccine. It's a subunit vaccine. Whenever the pathogens are very, I would say, highly dangerous, they would basically take out only specific carbohydrates or proteins specific to that pathogen, and they would formulate them into a vaccine and inject them. And so whenever you would encounter the real pathogen, your immune memory would recognize them based on specific proteins or carbohydrates. And then the last type, which is conjugated vaccines, is basically dealing with a weak antigen coupled to a strong antigen, the strong antigen trying to essentially induce an immune response against the weak antigen. And this is what adjuvants are essentially doing. So next I want to talk about what's inside a vaccine, just so you can understand what role adjuvants play. The first component is obviously the active component, and it can be live, heat killed, chemical killed in pieces, or lab engineered, as I mentioned before. Then you need a stabilizer, and this essentially ensures that the active component remains unchanged throughout transportation or administration of the vaccine, or storage of the vaccine administration. And this can range anywhere from a mixture of sorbitol to an inorganic salt such as magnesium sulfate. Then you can find traces of antibiotics, common antibiotics such as gentamicin or neomycin. They are removed supposedly, but you can find them in trace amounts. They are typically used in the manufacturing process to ensure no cross-contamination. Fourth component are preservatives, which are in fact active components, and they are most commonly found in multi-dose vaccines. These actually do ensure no cross-contamination. Other trace components you can find, and I mentioned before, formaldehyde is used typically in toxoid and inactivated kill type vaccines. And so those are the kind of vaccines that you would most likely find this compound in. However, it is present in such low amounts that it is not harmful. And then last but not least, you have adjuvants, and there are many different kinds, and they come in all shapes and forms, and I will talk about this in detail in a little bit. So we saw the role of all the other components, but why use adjuvants if you have an active component already present in the vaccine that can cause some sort of immune response and immune cell memory? Well, if you think about it, how much antigen can you put in a dose? You might not have enough antigen to produce as many vaccines as you wish, and in the case of mass pandemics such as we have right now, you may not be able to produce enough vaccines for the required amount of people. So adjuvants essentially enhance the immune response such that you are able to reduce the dose of antigen per vaccine, and you're able to form enough memory cells in your primary immune response such that the number of booster immunizations goes down as well. And how exactly do they do that? They are very good at recruiting antigen-presenting cells to the site of immunization, which will in turn lead to antigen recognition and cytokine production, therefore enhancing the immune response. However, like any other product, it has its good and bads. Can you in fact achieve potency while avoiding toxicity? Not so, and it depends on the type of adjuvant. Some of them may have only local reactions, and they can be anywhere from, they can range anywhere from injection site pain, inflammation, all the way to more severe local reactions such as ulcers and sterile abscesses. However, the more harmful kind are the systemic reactions, and these are the type of adjuvants that would be on clinical trials for an extended period of times or may never reach clinical trials because they can induce acquired immunodeficiency and immunotoxicity. So as you can see here, adjuvants can be classified based on type. They can be either delivery systems, immune potentiators, or mucosal adjuvants. And there are many more adjuvants that are not mentioned in this table. But I want to mainly focus on the ones that are either licensed or on clinical trials currently. And I want to start with aluminum salts, which are the most widely used. They're also combined with, they can also be combined with another type of adjuvant, monophosphoryllipid A, which is a gram-negative cell wall lipid, which by itself is not as great as an adjuvant. As you can see, it's still in clinical trials, even though it was discovered in the late 1970s. In combination with aluminum salts, it can prove to be a great adjuvant. And we will see that this is a pattern that repeats on with other types of adjuvants. You can also have oil and water emulsifiers, such as MF59 and Adjuvant System 3, which act similar to aluminum salts in a way. You also have other types of adjuvant systems, Adjuvant System 2 and 1, They are still in clinical trials. One is the liposome-based one, and one is an oil and water emulsifier. And they're both combined with saponins and monophospholipid A. Next, you can have virosomes, which are in the same category as virus-like particles. They are essentially viral glycoproteins combined with membrane lipids into viral envelopes. And then you can also have another oil and water emulsifier, which is the Freund's adjuvant. You probably have heard about it. It's quite common, but it is mainly used in the incomplete form, which does not contain the killed Mycobacterium strain. Next, we can see a pattern of adjuvants that use viral viral and bacterial RNA and DNA to induce an immune response, such as single-stranded RNA, imiquimod, resiquimod. Then you can have poly-IC, poly-ICLC, which is a double-stranded RNA adjuvant. You can also have single-stranded DNA, CpG oligodeoxynucleotide. And then you can also have different components from bacteria, such as parts of peptidoglycan or flagellin, which is a fairly common protein encountered in pretty much all bacteria. So why are there so many adjuvants? Can there be a perfect formula for an adjuvant that would work with every vaccine? Well, it turns out that each pathogen is different in the way that it needs to, each pathogen is different, and it requires a certain level of immune response in order to be neutralized and killed. And that brings me to the topic of how do adjuvants work. In this figure, you can see a summary of all the different types of immune responses that an adjuvant can enhance. And I will first start by talking about the type 1 and type 2. Type 1 being the cellular response and type 2 being the humoral response. As I mentioned before, the presence of antigen alone can, because it needs to be in such a small amount, it may or may not induce an immune response, and it is likely that it will not, especially because it's unable to replicate. So the dose that you inject is the dose that you have in the system. So it will most likely be recognized as not a threat. However, whenever you introduce an adjuvant, it can swing the immune response to recognize the antigen as a threat, because once immune cells are recruited over through pattern recognition receptors and immature dendritic cells, like once the immune cells come over, they can see the antigen attached onto the adjuvant, and then they can start considering the antigen as a threat, and it can launch two different immune responses. The type 1 is a cellular immune response, which deals with pathogens that replicate mainly intracellularly, and then the type 2 can be a humoral response, which deals with pathogens that replicate extracellularly. So the way this works is the antigen-presenting cells will basically engulf and not metabolize, but break apart the pathogen, the antigen or pathogen, and then they will display on their surface certain proteins specific to that antigen on either major histocompatibility complexes 1 or 2. And then you have T helper cells or CD4 naive T cells. They come over and then they read these immunological synapses, and they decide whether to induce a type 1 or a type 2 immune response. If they are to secrete interleukin-12, then the naive T cells will mature into T helper 1 cells, which will later secrete interferon gamma, and this will recruit cytotoxic CD8-4 cells, as well as natural killer cells and macrophages to the site of an infestation or infection. Interferon gamma can also cause B cells to proliferate and turn into plasma cells, which will later travel into the blood or lymph and then tag the pathogen with opsonizing antibodies. Opsonizing antibodies are essentially tagging the pathogen and recruiting more CD8 cytotoxic cells and natural killer cells and macrophages over to kill the pathogen. However, if the T helper cell decides to secrete interleukin-4 cytokine, it will mature into T helper 2 cell, which will secrete in turn interleukin-13, and it will cause the B cells to turn into plasma cells that will travel to blood and plasma and use neutralizing antibodies to tag the pathogens. These neutralizing antibodies are essentially used to stop the pathogen from replicating if it's a bacteria, or from infecting other cells in the case of viruses. The main point here that I want to make is that for some vaccines, you want either type 1 or type 2, or you may want both if the pathogen is very strong, because you want to be able to stop the cell to stop the pathogen before it infects a cell, and you also want to be able to stop to kill an infected cell if the pathogen enters it. And you also want to produce T and B memory cells. Next, I want to further divide adjuvants into damage-associated molecular patterns, or DAMPs, and pathogen-associated molecular patterns, or PAMPs. And I will start by talking about PAMPs, and this can either be can be categorized in two. One is the surface PRR pattern recognition receptor activation, and the other one can be endosomal. So antigen-presenting cells essentially contain these germline encoding pattern recognition receptors, and toll-like receptors are just a subtype of these PRRs that are able to detect pathogens both from the outside and the inside of the cells. And you will see that the PAMPs adjuvants essentially exploit these PRRs to their advantage. On the surface, you would have TLR1 and 2 and 6 that will recognize triacyl and diacyl lipopeptides, which are commonly found in the walls of microorganisms. You also have TLR4 that can recognize lipopolysaccharides, and TLR5, which can recognize flagellin. And in the case of a bacterial infestation, TLR5 would most likely be activated. And then you have the endosomal kind that are sitting inside the cell, inside the antigen-presenting cells, and these can recognize double-stranded RNA, single-stranded RNA, or single-stranded DNA. And these deal more with viral infections or pathogens that can divide intracellularly. Now notice that there's also any NOD-like receptors here, and I will talk about this in a little. I will talk about this next, but these deal with a different kind of adjuvant, and especially the damage-associated molecular patterns. And you can see here, it deals with the inflammasome formation and the caspase activation. And then I want to also talk about what happens when you combine damage associated with the pathogen-associated molecular pattern adjuvants. So alum is by far one of the most widely used adjuvants since its introduction in the late 1920s. This adjuvant is in the formulation of many licensed vaccines, such as in hepatitis A or B, or diphtheria, tetanus, and pertussis vaccine, and many, many others. It has, how does it work? It has a slow release because it precipitates, or a depot effect, because it essentially precipitates at the site of injection. And this allows for APC recruitment. And once it is taken up by the antigen-presenting cells, it will bind to what is called NOD-like receptor 3, which will in turn activate caspase-1 and the whole inflammasome system. And caspase-1 will produce one of the most commonly commonly known inflammation cytokines, interleukin-1 beta. In the case of some vaccines, alum is not enough because alum is a type 2 immune response adjuvant. And so sometimes it has to be used with another toll-like receptor adjuvant, which can induce, in fact, a type 1 immune response. And this is the case in many adjuvants. Now that we saw the different kinds of adjuvants and how they work to enhance the immune response, I would like to discuss a particular pathogen-associated molecular pattern adjuvant that has been of specific interest to our lab. And this is the CpG oligodeoxynucleotides. It's a class of synthetically produced single-stranded DNA, which contains a high proportion of cytosine guanine motifs, which are in fact unmethylated, and the reason it's a good adjuvant is because the mammalian genome contains less than 2% CG motifs, and they are typically methylated. In the case of the bacterial genome, CG motifs are very common and they are unmethylated. So this double-stranded, it's a single-stranded. This single-stranded adjuvant will take advantage of the toll-like receptor 9 pattern recognition receptors and will induce a pretty strong immune response. However, this has only been seen effective in vitro. Whenever scientists try to translate it in vivo, they realize that CpG can be highly toxic and non-specific. One thing that I forgot to mention about CpG, and that I think it's pretty cool, is that whenever you replace the phosphodiester bond with the phosphorothioate bond, you can make this molecule resistant to DNases, and this is what makes CpG stable in vitro. So, like I mentioned, it's non-specific and it's also can be cytotoxic. So, people have tried to combine it with another delivery, let's say, delivery system adjuvant, such as virus-like particles. Virus-like particles have this unique repetitive surface structure that makes them highly immunogenic and great vaccine templates. This rigid and repetitive structure facilitates B-cell receptors to cross-link, and this causes and facilitates antibody tagging as well as their uptake by antigen-presenting cells, which is subsequently what we want for CpG. However, one of the problems with VLPs is that they contain remnant bacterial RNA, but scientists have been able to to disassemble the capsid, remove the RNA with an RNase, load it with CpG, and reassemble the capsid. And in order to attach the antigen for a vaccine template, they have been using a SMPH maleimide linker that essentially rebridges reduced disulfide bonds on the surface exposed cysteines, and they will attach the antigen to that. So you would have the CpG inside and the antigen on the outside. The only problem with these delivery systems is that combining two adjuvants will essentially extend the clinical trials, but it can provide a better vaccine with better memory cells lifetime. So I want to briefly go over how clinical trials work, and it usually starts with preclinical trials, which is studies in animals. This will determine the number of doses that they need to form antibodies. will determine the site of injection needed, as well as what kind of immune response, type 1 or type 2 or both. Then once the preclinical trials are successful, you can introduce vaccines in the phase 1 into tens of healthy adults. And this will monitor for adverse effect that I mentioned earlier in the presentation, the safety of vaccination, and the other potential risks. If there are no severe adverse effects, then it will go into phase 2A. It will get administered to hundreds of target people. And this will monitor for more minor side effects, such as ulcers at the site of injection or inflammation. And they will also monitor immunogenicity further. If this is successful, it will go into phase 2 and 3, which is the last phase before manufacturing and licensing. And it will get administered into thousands of target people, and it will essentially determine if the vaccine is effective and safe enough to go on the market. And phase 4 is basically an after post-licensing and marketing stage in which they typically just monitor the safety and potential adverse effects of the adjuvanted vaccine. So this can take up to maybe 2 years. This is the reason why vaccines take so long to get marketed. But is it really worth it? Well, I would say so, because adjuvanted vaccines help increase antigen uptake by antigen-presenting cells. They also increase cytokine production and activate inflammasomes. They induce cellular recruitment and increase the rate of seroconversion, which is the amount of time that it takes for the antibody to appear in the blood. And all of these result in being able to use a lower amount of antigen per vaccine, and it also reduces the number of booster doses. And this also leads ultimately to a secondary immune response being induced more rapidly when you actually come in contact with the real pathogen. Thank you. That's all. Any questions?
That was quick. Okay. Anybody have any questions?
I had a question just about symptoms in vaccines. So some vaccines have symptoms of like muscle weakness or like tenderness in the shot area, or various things like that. But why is like that allowed compared to other vaccines that have no symptoms at all? Like, why do they allow those things, especially because vaccines tend to be a touchy subject nowadays? What is like influencing the amount of symptoms allowed?
I am not sure, but I would assume that you would have to deal with the level at which you're affected. For example, I mean, if you just have injection site, if you just have pain at the site of injection, that would go away in maybe a couple of days, then that would be assumed to be a safe adjuvanted vaccine. But if you would get an ulcers, which was the case with a lot of the Freund's adjuvant vaccines, Freund's adjuvanted vaccines, then you would have to be on clinical trials for either an extended period of time, or it's just not a good adjuvant and they should not market it. I'm not sure if that answers your question, but I'm sure there is a rubric of, obviously you wouldn't want to market vaccines that would cause systemic reactions, but if you're thinking about local reactions, I'm sure there's a rubric of like how painful they are and what are the side effects, and this is why they're on clinical trials essentially.
Yeah, I just think it's so interesting that like some are allowed and some aren't, even though like to me those are not bad symptoms, but when you talk about parents like not wanting their child to go through so much pain getting vaccinated and stuff like that, like it becomes like an issue. And like, how does the FDA like regulate that? And so interesting. more of like a discussion question, I guess.
Okay, anybody else?
Yeah, great presentation. I just had a question about the various kinds of adjuvants. You mentioned Freund's adjuvant, and I think you said that it's an incomplete adjuvant. What did you mean by that?
Yeah, so I mentioned that the complete form contains the inactivated or kill type of mycobacterium, while the incomplete one doesn't. And so they're using the incomplete one because it doesn't produce such severe side effects. But you can see that it was discovered how long ago, and it's still in clinical trials.
All right,
Yeah.
thank you.
Oh. Okay, anybody else?
You talked a lot about, well, there's a few slides, or at least one. You talked about this cells maturing. Can you please elaborate as to where?
I'm sorry, you're breaking up. Can you repeat the question?
Yeah. Can you hear me there?
Yeah.
Okay, so you talk a lot about cell maturation before it's sent to fight the pathogen. Can you kind of elaborate as to exactly that? Can you please elaborate as to where is this process taking place in the body? If that's something that was included in your presentation.
From my understanding, it happens in the lymph node. I think naive T helper cells will go and process this in the lymph node, but I'm not 100% sure.
Okay. Normally, okay, yeah. That was it. From my understanding, I think it happens in the bone marrow, but I just wanted to know if there was a different place where it could take place as well.
See, I thought that they get produced in the bone marrow. So like T cells will go and mature in the thymus, and then they will just go roam around in the bloodstream, but
I see.
But yeah, I don't think you
Thank
want
so much.
cytotoxic cells just circulating in your system unless they're activated. So
Thank you. Great presentation.
thank you.
Alicia,
Oh, hi, Michael.
how you doing? Great talk.
Thanks.
I guess a question I had is, can you give an example of what virus or bacteria would be better for a type 1 response and what would be better for a type 2 response?
So for sure, type 2 would be virus or bacteria or adjuvant.
I guess I wonder, like, in all cases, wouldn't it be better to have either one, occur?
Yeah, that's why I said I think by combining two different kinds of adjuvants, they're trying to achieve that. Hold on, let me go this way. Yeah, so they're trying, for example, aluminum salt will give type 2, while this will give a type 1 or something. Like they're trying to combine two different adjuvants that will produce both types of immune response. But essentially, type 1 is cellular, so it will mainly target pathogens, viruses, right? Because they divide intracellularly. And then type 2 will mainly target bacteria. But I'm seeing a lot of recurring pattern with adjuvants where they're using both kinds to induce both types of immune responses.
Okay, thank you.
Mm-hmm.
So remind me, CPG is type 1, right? Or is it type 2?
Type 1, intracellular.
Yes. So that would be a pretty good pretty good at these cancer vaccines, I guess, then, right? So I know this was beyond probably what you were looking for, since you were looking for disease, but did you see anything with CPG if like the cancer immunotherapy as an adjuvant?
Yeah, I've seen some papers, but I haven't really dug into it. But I can look more into it.
No, this was very good, very complete. I really like this talk. Anybody else? Yeleni, have something?
It's a really nice presentation.
Thank you.
Yeah. Okay, well, if that is all, then let us all congratulate Alisia on a good talk. Clap in front of your monitor.
Good job, Alisia.
Thank
Good
you.
job.
Very good.
Great presentation.
Yeah, that was great.
Thank you so much again, Alicia.
Yeah,
I
yeah.
can only imagine the amount of reading you have to do. Thank you.
Yeah, that was probably a lot of reading. You should maybe consider doing research.
Oh my
Becoming a
gosh.
doctor, at least. No, I kid. All right. Is there a talk on Friday? No, we're not doing Fridays anymore, are we? Okay. All right. Well, then I will see you all on Monday. Okay. I think. Bye.
Bye.
Wait, isn't my talk?
Yeah, Wednesday.
My talk's supposed to be Wednesday, so he doesn't want to do it.
I think he thinks today is Wednesday, since he immediately jumped to Friday.
He's gone.
Okay.
We'll be here.
Happy birthday, Laurel. Is she still around? Oh yeah.
Yes, I'm here. Oh, thank you.
Are you 24 now?
No, 23.
23. Okay, well, hopefully we'll get back to normal
Where
as
did
soon as
you-
possible, and we will take you all out to eat something.
Yeah.
Say
Take
something.
Indian
That's totally
food,
saying
please.
happy birthday.
I'm like slouching in my
Is that what that face was?
That's okay. You don't have to sing. It's fine.
No, I will sing for you. Let me go get some fire as your fake.
Fire. What? Oh gosh, should I just leave now? Well,
I'm gonna like. See, it's like your birthday candle. Except it's just hemp wick.
Oh my God.
Candle.
right now. What is that?
It's your birthday candle.
Aw.
Oh, fire, it's for you.
Shava,
Hey.
how you doing?
I'm good. How are you guys?
Oh my
I
god.
miss you so much.
Hi. It's been so long.
On my
I wanted
back.
to let you know,
Oh my God.
guys, my flight was supposed to be on the 4th of May 4th, but they changed it and they keep changing it, and now it's on the April 30. So it's like in 3 days,
Thursday?
and I might be going back like on Thursday. But I'm
Please
still have
tune in for
internet
my
in France. I
presentation.
will come to the group meetings and everything. So yeah.
Oh well, Jeremiah just sent me though about
Yeah.
the meeting on Wednesday. Oh well, I'm glad you're getting to go home because I feel like this was not the experience we thought like you were gonna have.
Yeah, I mean,
like come to America and be cooped up.
yeah,
But
but I still had fun with you guys, with my family, and yeah, I was mostly with my fans, but they're here now, and yeah, I can't wait to see them, even though I won't be able to hug them. But anyway,
yeah. Oh, that's good. I'm glad you're at least like allowed to travel and go home.
yeah, they kept changing my flight. They kept getting canceled, and now it's on the 30th. So we'll see if it will be canceled or not before. But I hope it's going to maintain.
Yeah.
Laura, you ready for this?
Oh, is this still happening? I was trying to
Yeah.
conversation away.
Happy birthday, Laurel.
Yeah, happy birthday.
Sing it.
How many people are still in here? An 8-part duet. Or
I don't know about an 8-part duet with internet lag. You're asking a lot.
an 8-part
Heard
harmony.
the lab sing happy birthday before? You gotta know.
What did you?
Have you ever heard the lab sing happy birthday?
No.
Because I was gonna say, if you've heard it, then asking for a 2-part duet is even a lot.
My family likes to sing it as off-key as possible, so it's really bad every time. And then my mom tries to sing happy birthday on the same note. And like, it's horrible.
Okay, I will at least give you a good one.
I'm ready.
Happy birthday to you. Happy birthday to you. Happy birthday, dear Laurel. Happy birthday to you.
That was beautiful.
You're worried about me singing all terrible.
Tears. I need to get some wine in me. I'm like still celebrating, but kind of not. But
You're still what?
I'm like still celebrating my birthday, but kind of not because I'm also doing work now. But
Anybody want to finish my thesis?
I'm having a hard enough time with this lab report, semester report, whatever it is.
Yeah, I'm not writing that, so.
Oh, must be nice.
Yeah, but I have to finish my thesis.
It's basically just putting a lot of detail in the writing about how all of your experiments didn't work. So it's a lot of fun.
I know. Mm-hmm.
And then having to come up with like ways that you're going to improve. Yeah, it's great.
Yeah, how you're gonna fix it, and you're like,
I have no idea. When will I be able to fix it? Who knows? Okay, I gotta go. Thank you guys for the belated birthday wishes, and I'm looking forward to celebrating in person whenever that can be.
yeah, let's just hope it's before I get kicked out of the lab.
I hope so. Well, you already said you're not even coming back to Texas, so.
I mean, technically, no, I am coming back to Texas. Like, I'll be back to Texas this weekend. As LOL, when I came here, I didn't have my license, and my passport expires on May 3rd. So I need to be back in Dallas.
Yeah.
But I mean, I'll be back. It's just whether Jeremiah is willing to pay me to be back or not. That's really the hang up. Can't live without pay.
Yeah.
Kids, go do something productive with your day.
Yeah, we'll do.
Especially you, Yeleni, go outside.
Bye. Really good talk, Alicia. I loved it. That was so good.
Nice talk, Alicia.
It was okay.
Thanks, Candace. Love you too.
All right. Bye, ladies. And Nima.
Hi, Dora.
Bye, everyone.